Hello, my name is Julianna Bourgeois, and I'm a graduate student in the Interdisciplinary Biological Sciences program at Northwestern University, where I work under the guidance of Professor Chad Mirkin. Today, I'd like to pose a question. What if the way that we discover and deliver medicine is about to fundamentally change? I'm going to tell you about a revolution in drug development and how researchers at Northwestern and the International Institute for Nanotechnology are leading the way. Over the next few minutes, we'll explore how a new field called structural nanomedicine is redefining what success looks like in this space. Furthermore, this new approach will dramatically change how we treat the world's most debilitating diseases, from cancers to infectious diseases, neurodegenerative disorders, and many more. If we were to look back just 10 years ago, we would find that the drug landscape looked strikingly different from what we see today. At the time, many of the top-selling drugs were small molecules with familiar names like Crestor, Lyrica, and even aspirin. But today, the most advanced drugs are no longer just small, simple molecules. Instead, drugs have evolved into larger and far more complex structures, growing from biologics to gene therapies, and even into highly complex and sophisticated nanostructures. Today, many of the most powerful drugs are made from short snippets of biologic material like DNA, with some even being called miracle drugs for the way they've transformed once incurable diseases. Take Keytruda, for example, which has dramatically improved survival in cancer patients, or GLP-1 drugs like Ozempic and Mounjaro that are now reshaping the treatment of diabetes and obesity. But perhaps no breakthrough has been more globally recognized than the COVID-19 mRNA vaccines, like those from Pfizer and Moderna. These vaccines use tiny lipid nanoparticles to safely deliver fragile mRNA molecules into our cells. These nanoparticles essentially act like protective bubbles, shielding the genetic instructions until they reach the right place in the body where they can trigger an immune response. Simply put, without this nanostructure, the vaccine would not work. These new complex nanostructure formulations mark a turning point, as they show us that moving beyond traditional small molecule drugs can lead to truly transformative results. The fact that such medicines can now be developed quickly and effectively also signals that the industry is ready to welcome in a new era. Still, with this shift comes new challenges. To unlock the full potential of these new drugs, scientists must learn how to assemble molecules into precise, well-defined structures that are not only safe, but remarkably effective. And this is no small task, considering that when working with multi-component nanostructure systems, the number of possible combinations grows exponentially. However, not every design structure is going to be effective. To use an analogy, building a drug is essentially like assembling a car. There are thousands, if not millions, of ways to put the parts together, but I think we can agree that the version that has the wheel on the ground is going to work a lot better than the ones where the wheels are on the roof. Now, if we were to imagine making a nanostructured drug made up of three components, say a nanoparticle carrier, a piece of genetic material, and a small protein snippet, with each component itself coming in three different types, the number of possible particle combinations is equal to 27 unique formulations. However, if we further consider sequence, backbone, lipid type, anchor, peptide positioning, and consider arranging all of these different components into different orders, the number of possible arrangements grows dramatically, with a number of potential outcomes numbering well into the millions. Within these millions of potential designs, there lie novel structures that can either be completely ineffective or curative, and finding the strongest candidates among this vast set of possibilities is a huge challenge. This is why researchers are now employing AI and machine learning techniques to pare down the enormous number of possibilities into a manageable set, enabling the discovery of the safest and most efficient designs. The field of structural nanomedicine, where drugs like the COVID-19 nanoparticle vaccine are optimized using precisely engineered nanoparticle platforms, champions the premise that rigorously investigating a manageable set of structures made up of different drug components can unlock new therapies with maximal efficacy and minimal toxicity. With researchers at Northwestern and the IIN working tirelessly to build and drive the progress of structural nanomedicine, they have established us as the global epicenter of this effort. Indeed, many researchers at Northwestern are developing structural nanomedicines across the fields of chemistry, material science, and biomedical engineering. Notable examples include the spherical nucleic acids pioneered in my advisor Chad Mirkin's lab, are being developed as therapeutic tools to perform gene editing and treat cancers and infectious diseases. Another example are Milan-Merck-Sich's megamolecules, which are atomically precise protein structures that are employed as diagnostics and therapeutics. Beyond these examples, other leaders in this area include Professor Shana Kelly, Leon Platanius, Ali Shalatafard, Sam Stoop, Bin Zhang, Doug Vaughn, Bob Langer, as well as current and past Kabiller winners, all of whom are utilizing the principles of structural nanomedicine to revolutionize the therapeutic landscape. Much like how Northwestern has capitalized on nanotechnology in general, clean energy initiatives, and the Chan Zuckerberg Initiative in immunology, we are now poised to spearhead a massive global effort that will lead the way in pioneering the landscape of structural nanomedicine. These efforts and advancements require collaborative and multidisciplinary efforts made possible by generous donations from organizations and people like Ed Bachrock, Ray Nimrod, Doug Gray, Tim and Sue Sullivan, Patrick and Shirley Ryan, and others. I would also like to thank Lee Beenan for all of her help in enhancing scientific communication. As a Ryan Fellow myself, I am extremely grateful for the funding and support that allows me to contribute to the field of structural nanomedicine, a field that will undoubtedly impact the medical world and one that has the potential to positively impact the lives of millions of people. Thank you.